then Mr. Samir Moirangtem, Assam Downtown University, will be talking on the emergence of multi-drug resistant and biofilm forming escape pathogens from clinical, environmental and food sources in hospital settings of Mizoram.
A very good morning to each and everyone. So today I'm going to present emergence of multi-drug resistant and biofilm forming escape pathogens from clinical, environmental and the food sources in hospital setting of Mizoram. So introduction. Escape group, it includes Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa and Enterobacter species. Why it is very major concern? These bacteria, they can escape the effect of many antibiotics, so that is the major concern. Global health challenge, rapid rise in the antibiotic resistant, treated effective infection controls we needed. And hospital impacts, they are responsible for the large share of hospital acquired infection and high morbidity rate. So beyond hospital, recently found in food sources like example of meat samples and then many samples shows spread beyond the healthcare settings. Cross-contamination risk indicates possible transfer from hospital and the community. So the purpose of my study is comparing the prevalence of escape pathogens in clinical specimen, environmental and the meat samples. So objective of my study is this study aims to explore the prevalence and potential sources of escape pathogens within hospital environment with a specific focus on any linkage with the meat that is sold in and around the market of Mizoram. So for this research, we took the ethical clearance obtained from the IEC refunds and required permission were obtained from the hospital and the consent were taken from all the participants. So altogether, we collected around 200, we collected samples of 200 samples, and from the clinical samples, we collected 100, including bowel, tracheal aspirate, urine, blood specimens. And then from the meat sample, we collected the chicken, pork, beef, fish, and mutton. From environmental, we collected from the wards, OPD, kitchen, operation theater, emergency, and the ICU. So the methodology. The sample collection, food meat samples collected aseptically from different sources. The sample processing, homogenized and enriched for the isolation. Laboratory diagnosis, we culture on selective and differential media. Morphological examination, we perform gram staining and colony morphology. And for the biochemical identification, we did IMBIC test, catalase, coagulase, urease, etc. So for the antibiotic sensitivity testing, AST test, we use Kirby-Bowers disc diffusion method. And for MDR determination, we perform Congo-Red, agar, and tissue adherence method. And for the stock preparation, we use 20% glycerol, and we did the statistical analysis. So these are the antibiotics I used during my research. So these are the coming to the finding. So overall, this is the prevalence of escape pathogens in hospital, clinical, and the environmental. So the majority, the Staphylococcus aureus shows the highest prevalence of 36.3 and the least Enterobacter species. And the second one largest is shown by Klebsiella pneumonia with 27.2%. So prevalence of escape in clinical samples, urine shows the highest positive rate, which is followed by pus and then tracheal aspirate and then bowel. So from the clinical aspirate, Staphylococcus shows the highest prevalence with 41%, which is followed by Klebsiella pneumonia with 34%, and the Pseudomonas aeruginosa 16%, Enterobacter 3%, and then Acinetobacter baumannii 6%. So again, the prevalence of escape in meat samples. In meat sample also, surprisingly, the chicken shows the highest prevalence, and the pork, beef, fish, and mutton. Among these different samples, chicken shows 35.5% of contamination with escape pathogens. So from the meat samples, Enterococcus faecium shows the highest prevalence with 36%, which is followed by Staphylococcus aureus 29%, which is again followed by Acinetobacter baumannii with 21%, and the last but not the least, Klebsiella pneumonia also showing 14%. Coming to the prevalence of skin pathogens in hospital environment. So in the hospital environment, the emergency shows the highest prevalence, which is followed by the ICU. Actually, ICU is supposed to be very sterile, but during our studies, it shows very contaminated. So from the hospital environment, then Staphylococcus, again Staphylococcus aureus shows the highest prevalence with 35%, followed by Klebsiella pneumonia with 25%, and Pseudomonas aeruginosa 5%, and Acinetobacter baumannii 10%. After this one, we perform the AST patterns. This is the antibiotic patterns of escape pathogens isolated from clinical, hospital, and then meat samples. So these are the resistant antibiotics. So from this one, the cepism shows the highest resistant, and then which is followed by the PIT. So coming to the biofilm formation, total we isolated 66 number of escape pathogens from altogether 200 samples. From this one, 61.1% are forming biofilm, which is very concerned. So among this one, again Staphylococcus aureus formed the maximum number of biofilm with 36.7%, which is followed by Klebsiella pneumonia with 24.3%. And the least it is found in the Enterobacter species, mainly Enterobacter aerogenes and Enterobacter cloacia. So we classified biofilms into three types: weak, moderate, and strong. This is we classified based on the optical density control value. If it is less than if it is less than 2, we take as none or weak. If it is between 2 to 4 ODC value, we used to take this as the moderate. If the ODC value is more than 3.5 to 4, we consider that is the strong biofilm organism. So coming to my discussion, escape group, all this Enterococcus faecium, aureus, Staphylococcus pneumonia, Acinetobacter, Pseudomonas aeruginosa, Enterobacters, they are mainly not only found in the hospital environment, they are also found in the meat sample and even in the clinical samples. So major concern, they are key contributors to the global antimicrobial resistance, and then their unique feature, they can escape the action of the antibiotics. So my study focus examined spread of escape pathogens across clinical, environmental, and the food samples. And the study location we conducted in the hospital Benicities in Mizoram to trace transmission route. So key finding, the most prevalent isolate is Staphylococcus aureus, followed by Klebsiella. So they are mainly found in the urine and the wound specimens. Next common pathogen is Klebsiella and Pseudomonas aeruginosa. The major global comparison is aligned with the world data, that is Staphylococcus aureus shows the highest prevalence in urinary and respiratory tract infection. So finding from the meat sample, meat products near hospital shows bacterial contamination. Main isolates are Enterococcus faecium, Staphylococcus aureus, and the possible cause during the slaughtering of the meat or because of the spillover from the intestine, they may be contaminated. So from the antibiotic resistance, the carbapenem shows more than 98% resistant, and then 88% are resistant to penicillin derivatives. So for the biofilm formation, many isolates shows strong biofilm producing ability. Biofilm enhance the ability to antibiotics and disinfectant. So that is the major concern why I choose this topic. So high prevalence is the organism pathogens, they are found in the ICU, surgical wards, and the general wards, and then major contributor to the hospital acquired infections. So antibiotic, that can be because of the overuse of the antibiotic or the misuse of the antibiotics, and contributing factor can be immunocompromised patient, invasive medical procedure, and poor infection control practice. So in food chain linkage, not only this escape pathogen, they are also found in the meat samples. So comprehensive study, this study reveals how escape pathogen extend beyond hospital setting, not only in the environment, not only in the clinical. Thank you. Yes, any question? Yes, sir.
In this study, you have collected samples from various sources.
Pardon, sir?
You have collected the samples from various different sources.
Yes, sir.
Clinical samples, then meat samples, environmental samples.
Yes, sir.
Did you find any correlation between these all sources, and you found that the organisms present in environment or organisms present in meat, mutton, chicken, those were responsible for causing some infections?
Yes, sir.
This is requiring epidemiological typing.
Yes, sir.
So without epidemiological typing, you cannot blame any organism.
Sir,
So
but yes, sir.
on what grounds you are saying that these are important organisms?
Sir, why I specifically took this escape organism, escape pathogens, because they are the leading organism that is causing host nosocomial infection. So since this nosocomial infection, they have the ability to escape many actions of the antibiotics. So they are also found in the hospital environment, even they are also found in the meat, but the people, the normal people, they doesn't have that much idea about the hygiene practice. So what I want is, I just want to try to find out the main root cause or the sources of infection, whether these escape pathogens are mainly infecting or originated from the hospital or from the meat sample or from the clinical. But as of now, till now, my further studies needs molecular level, so I'm planning to go for the molecular. But as of now, this is only the preliminary screening, sir.
Yes, you have worked hard, but I think it would have been a much better study if you could have linked these organisms, or linked these sources.
Yes, sir.
That these organisms which have caused infection in patients,
Yes, sir.
those organisms have come from either mutton or from chicken or from the environment.
Yes,
So
sir.
the correlation, epidemiological correlation is very, very important.
Yes,
Without
sir.
that, I think we cannot blame these organisms.
Yes,
So
sir.
you can go ahead for further studies.
Yes, sir. Sure, sir.
Okay, you should
Yes, sir.
go for epidemiological marking of such organisms available from. You can go for mapping from the organisms isolated from the environment or other sources, and the organism isolated from clinical samples, whether the organisms are same or are different.
Yes, sir.
That's very, very important. Otherwise, if you say that drug resistance is there, that is all that everybody knows. Drug resistance is there in these organisms. Pseudomonas, acinetobacter, staphylococcus.
Yes, sir.
Okay.
Sir, but in that case, like if we can find the root, like the people who are staying in the hospital, who are already admitted in the hospital, for them, there is chance to get infected. But those persons who are not being admitted in the hospital and those who are taking from the butcher or anyone, even they also get infected with this organism. So my concern is about, I just want to focus this because they are the main leading organism that cause nosocomial, plus finding this organism in the meat samples also implicated our future concerns, sir. Thank you, sir.
Anyway, any questions from the audience?
Any questions? Okay, thank you.
The next talk is on clinical utility of biochemical parameters in IEEM Screening A retrospective study by Ms. Shambhavi Shetty. MLT, Manipal College of Health Professions, Mahi, Manipal.
Good morning everyone, I am Shambhavi from Mahi Manipal. I am here to present a study titled Clinical Utility of Biochemical Parameters in Screening for IM, a retrospective study. And this is our team. So, going with the background of IM, it is a group of disorders over 500 and it is a genetic disorder while each of them is individually rare, but their combined incidence is very higher. So, it resulting from the disruption in the metabolic pathway caused by the deficient activity of enzymes or the transporter molecules or the protein molecules involved in the biochemical processing. So that leading to the deficiency and causing the toxic metabolite accumulation, impaired energy production, contributing to the clinical manifestation of the disorder. So the global report of prevalence of this is 50.9 per 1 lakh live birth, and globally IM account for estimated 3 deaths per 1 lakh live births. So in higher income countries, the case fatality rate is about 2% to 23%, whereas in the middle income countries it is around 33%. But there is no reliable reports from the low income settings, primarily due to the limited availability of the diagnostic facilities and inadequate reporting system. So with that background, we come up with the aim of evaluating clinical utility of the biochemical parameters along with the clinical presentation for early screening of various types of IEM in the pediatric population of 0 to 3 years. So this was our method. So we have retrospectively analyzed from the time period of 2017 to 2023. We have selected only clinically detected cases, and we have extracted the relevant clinical features and biochemical parameters among these patients, and then we have analyzed the data with the random forest and lasso regression. So the study considered all the cases of IEM presented during that period of time, and total of 130 new IEM cases were found during that period, and we have followed international classification of inherited metabolic disorder for the classification. So this was the frequency of different IM in this study cohort, which was predominantly by the 55% by the energy metabolism disorder, then rest 35% by the general IM or unclassified IM, and followed by the amino acid disorders and uric acid disorders and others. And this was the clinical presentations presented in this study cohort. Some presented with isolated symptoms and some presented with the multiple symptoms. So general symptoms such as vomiting and other were higher, and followed with the respiratory symptoms and gastrointestinal symptoms. So then machine learning models such as random forest and lasso regression were applied to identify key biochemical parameters which can predictive of IEM and to classify IEM from the energy metabolism disorders. So the random forest model enabled high dimensional classification and determination of variable importance, while lasso regression provided a robust variable selection through regularization, preventing overfitting and improving interpretability. So this was the result of the random forest model by using two matrices, mean decrease accuracy and mean decrease Gini, and here the dots represent each of the variables or each of the biomarkers which we have used. So bicarbonate and lactate was the predominant one here. Energy metabolites such as lactate, pyruvate, lactate to pyruvate ratio, bicarbonate are the top contributor consistent with the known IEM pathophysiology and their disruption in the mitochondrial dysfunction and energy metabolism disorders. Overall in this cohort, lactate to pyruvate ratio was more in 99% and 1% exhibited reduced ratio. And when we have compared the feature of both random forest and lasso regression, we have got the results. Lactate and bicarbonate again topped the list, and pyruvate and oxygen content also had an impact. So both models constantly identified lactate and bicarbonate as the strongest predictors, and these findings reinforced the biochemical relevance in the acid-base and energy metabolism marker as the early diagnostic indicators. So the random forest helps to identify subtle non-linear contributions from parameters like methemoglobin and pyruvate. Coming to the discussion, the finding from this study provided evidence regarding frequency of energy metabolism disorder in our cohort, and compared to international data and observed frequency in this cohort underscores both the need for expanded diagnostic infrastructure, value of population-specific studies which can account for the India's genetic and environmental heterogeneity. And when we have integrated these markers in the pathway analysis, we have got the relevance in the various pathways such as glycolysis, gluconeogenesis, citric acid cycle, pyruvate metabolism, and amino acid metabolism. Also, we have found the relevance of ABG parameters in the mitochondrial dysfunction. So, whenever there is a mitochondrial dysfunction, even with the ABG parameters normal, there is a lack of oxygen utilization which can lead to the anaerobic glycolysis, leading to the lactate accumulation. and which requires bicarbonate for the maintaining pH stability, buffering the lactic acid generated during that impaired mitochondrial dysfunction. So shift in lactate and bicarbonate level often signal mitochondrial dysfunction, metabolic acidosis, onset of cellular stress, making them reliable early indicator of systemic metabolic imbalance. So machine learning enabled identification of these clinically relevant biomarkers by integrating complex biochemical data and uncovering both linear and non-linear relationship. So this approach supports not only early and precise disease detection, but also deeper understanding of disease mechanism, guiding targeted intervention and personalized management. So concluding with, combining random forest and lasso regression provided a rigorous data-driven framework for identifying robust biochemical markers in our inherited metabolic disorder cohort. So novelty of this study is prevalence data on the energy metabolism disorders in the Indian cohort. Recent genetic analysis also underscores the significant genetic diversity within India, so it requires more and more of prevalence studies. And the present study also addresses important knowledge gap by systematically documenting frequency and characteristics of energy metabolism disorder within this cohort, also in the Indian cohort. And the novelty of this study lies in the application of both random forest and lasso regression to the well-defined IM cohort, enabling rigorous cross-validation of predictor biomarkers and enhancing biological interpretability. So concluding with, biochemical screening can be a bridge between the clinical suspicion to molecular diagnosis, providing easy and cost-effective for the patient, minimal instrumentation technique, and further guiding the confirmatory testing. Thank you. I acknowledge the support from my department and institution, and thank you for the opportunity.
Thank you, Ms. Sambhavi. So, in your study, what is the most common disease you get about inborn error of metabolism? What was the most common disease?
So most common was energy metabolism disorder, energy deficiency for their developmental growth. So there is more of developmental delay or trunk muscle abnormality, all those cases.
Have you got any glycogen storage diseases?
One, only one.
Only one.
Yeah,
Glycogen
carbohydrate
storage.
metabolism disorder was there, but it was one.
So, for this IEM, what are the markers you advise, what are the markers you do regularly for this study?
Sir, there is a 2 or 3 type of tire testing. So first tire testing includes metabolic workup such as lactate, pyruvate, ABT screening, and also there is something CBC, all those things. And second tire testing includes confirmatory testings like amino acid metabolism, like amino acid profiling, and many more, GCMS analysis, LCMS, and confirmatory includes molecular diagnosis and genetic abnormality identification.
So for confirmation of these cases, have you done any genetic testing or any molecular testing?
Yeah, few of them had a genetic testing done, but not in all the cases.
So only screening test you have done for IEM?
Yeah.
Okay. Thank you.
Thank you, sir.
So what is the conclusion you are saying that by detection or early detection of this inborn errors of metabolism, so we can detect early what is that metabolic defect?
Ma'am, actually 1 to 2 years or 0 to 2 years is the developmental period. So during that time, if we detect earlier and provide intervention, it can be treated and it can be avoided, developmental delay or other things. So during that time, mitochondrial disorders, if treated early, it can be good. And also there is something called, if we go directly for the confirmatory testing or for the other one, it is costly for the patients. Also sometimes it takes time and more the things. So we want something which is cost effective, minimally invasive, also non-invasive also. We are working on that also. So something which is easier and something which is easy to adapt to the Indian cohort. So that can be easier. So like
After treatment strategy, you have the follow-up for the cases?
yes.
You have the follow-up?
Yeah.
Thank you.
Thank you. Thank you, ma'am.
Dear delegates, I have an announcement that if there is any of the presenter who has not submitted his or her PPT on the podium, please do it. send your PPT to the podium, to the comparers. Submit it immediately. Thank you. We may continue.
I study on lipid profile and anthropometric parameters among fasting people in Saharsa city, an urban area of Bihar.
Good morning, respected chairperson, faculty members, delegation, and my students. Myself, Professor Dr. Arjun Kumar, Professor Head, Department of Medical Lab Science, Narayan Medical College, Saharsa. My study is a study on lipid profile and anthropometric parameter among fasting people in Saharsa city, an urban area of Bihar, India. Background: history of fasting goes back thousands of years, including Muslim community. Each group of people use fasting as a healing process to cure sickness. Physicians, philosophers, and scientists believe that fasting affects not only our physical being but also our mental, emotional, and spiritual health. Fasting is promoted to regulate the body, help maintain normal body functioning, increase longevity and sex drive, and improve mental clarity, self-awareness, and self-esteem. It is said to be helpful non-prescription drug, so in few cases, instead of being beneficial to health, even a short term, and can produce a negative impact. Through understanding this study by the way of laboratory investigation, the data and result outcome of the study will play an immense role in health promotion and prevention of disease. This is a hospital-based study considering a small group of population in Saharsa city. Aim and objective. In this study, we will find out the effect of fasting on lipid profile and anthropometric parameter among healthy fasting people. Material and method. The study consists of 100 healthy people, 60 male and 40 female. In the age group of 15 to 65 years was carried out to biochemical test and anthropometry BMI, body fat parameter is done at 3 different times. Pre-fasting 1 week before the Ramjan, during fasting mid of Ramjan, and post-fasting 1 week after Ramjan. During the period February 2025 to April 2025, this in Saharsa district, Bihar, India. Anthropometric parameter which is used in our study is weight, was measured in the nearest 0.5 in pound. Height was measured in the nearest in 1 inch, and body fat percentage. BMI was calculated by dividing the body weight in kilogram as 1 kilogram is equal to 2.220 pounds by the height in meter square as 1 meter, 39.3701 inch. In biochemical parameters which are used in this study is lipid profile, cholesterol, normal range 135 to 200 milligram per deciliter, the serum triglyceride in male 60 to 170 and female 40 to 140 milligram per deciliter, high density lipoprotein cholesterol 30 to 70 milligram per deciliter, low density lipoprotein cholesterol 63 to 129 milligram per deciliter, very low density lipoprotein cholesterol VLDL to 41 milligram per gram. Statistical class analysis, the basic description, study, statistics and pair t-test used to compare the pre and post statistics on lipid and anthropology parameter. The correlation coefficient was work out to find out the degree of association between anthropometric parameter on the one side and lipid fraction on the other. This is the percentage distribution age and height. In male number 60 and below for 25 years, 31, 25 and above 29 age, below the average 35 and above the average age 25. In female, which are counting number of 40 females patients, below 25, 20 and above 25, 20, below the average 60 and above the average 24. The description statistics of age and height variable, mean study, height and age, mean or standard deviations both male and females. And this is find out before Ramzan and during Ramzan, after Ramzan, they calculated the BMI and fat which are reduced after during the fasting mode. So I think that the male and females BMI are reduced before before Ramzan and during Ramzan and after Ramzan, changing them at 1.93 and 1.12, and fat also decreased 27.45, that is percentage released to 1.80, and female the percentage 0.64. This is the interconnection matrix of anthropological biochemical profile before Ramzan, during Ramzan and after Ramzan. The parameter is this, and before Ramzan 1.0 BMI, and during Ramzan mid fasting 1.0, and after Ramzan 0.97. In the fat, cholesterol, triglyceride, that is also parameter is mentioned there. The result and conclusion, the major finding of this study are fasting having its impact on the BMI and fat, as from duration pre-fasting to the mid of fasting, there are negative change, and middle fasting to post-fasting having positive change in both the male and female, that indicate positive response of fasting on anthropological function of the body. Lipid profile also having similar results between male and female. There is a significant difference between the change in anthropological and biochemical parameter during the period of pre-fasting to mid-Ramzan and mid-Ramzan to post-fasting. BMI and fat and cholesterol and LDL are highly associated. According to my study, the body needs to get up and go to free the fat after fasting. The data and results of the study will engage the recreation and diagnostic task in anticipation of disease and health support. Thank you.
So you want to study the anthropological parameters with lipid profile. Significant results you have reached?
By the way of my laboratory investigations, by the way of biochemistry analyzer, automatic, which taking sample collection by the way of camp in the Muslim minority area, and collect the sample and analyze in automatic analyzer, then clarify number of patients come and contribute who have taken proper fasting during Ramzan. I hope in the Muslim minority, Ramzan is a holy fasting properly way, and I think this is a very important role during fasting. They decrease the fats according to. So we are taking clinical lab investigation. Cholesterol and fat is in our body which are reduced. So according to lab investigations, fully automatic analyzer will operate, and hospital authority will give permission for that. You will do the research papers on this very good, and I think I hope I will take the large number of patients in future for this year. According to the fasting related research will be do how paramedics or biochemical parameters will be decreased, and I think all who are the audience also take the sample in fasting mode, take better report, better results.
I'm asking, out of the samples you have taken, what is the significant results you have achieved?
Yes.
Thank you.
Thank you.
To talk on the presence of FKS gene and its association with multi-drug resistance in Candida species. Ms. Srimohi Shom, Faculty of Paramedical Sciences, Program of MLT. She'll be talking on the bacteriological analysis of mobile phones, a cross-sectional institute-based study.
Respected judges, my dear fellow guests and everyone present here, I am Srimohi Shom, a student of Assam Downtown University, currently pursuing BMLT. So today I am on behalf of my group research, going to present it. So our work was carried out by myself, Shaheen Akhtar, Sheikh Afifa, Sabina Sultana, Pelteng Jamatia, and Takelai. So I would like to begin with a question. If our mobile phones could show everything that lived on their surfaces, would you still hold it so close to your face? Probably you would not. You would hesitate after knowing what's actually present there. So, beneath that sleek glass surface lies a microbial fingerprint, a reflection of our hygiene habits, our surroundings, and sometimes our negligence. So this idea was the basis of our study, that is bacteriological analysis of mobile phones, a cross-sectional institute-based study. So these are the contents. That we'll be discussing today. So with over 4.7 billion mobile phone users worldwide, phones have become an indispensable part of our daily as well as professional lives. But despite being handled so constantly, they are rarely clean, which makes them a high contact foment. Around 80% of the infections are spread through hand contact. And in that case, mobile phones act as a bridge in the chain of contamination. Some studies have also shown that phones can harbor a variety of bacteria such as Staphylococcus, Pseudomonas, E. coli, and even some viruses like influenza, SARS-CoV. Here is a diagram showing how phone can be a link between these contaminated places and our body. You see, people use phones in various places such as washroom, cafeteria, from where they collect the microbes on it and then it spreads to us. So in this study, our main objective was to assess the bacterial contamination present on the mobile phones used by the students of Assam Downtown University and to understand how personal habits may contribute to the microbial spread. So our main objective was to identify the types of bacteria present on the mobile phones, to assess the usage behavior, and to evaluate the hygiene practices among the students. So how did we do it? We conducted a cross-sectional study. We included 40 student volunteers across different departments of our university, all were regular phone users. And then it was conducted from February to May of 2025. And then, under aseptic conditions, we used sterile cotton swabs. Those were moistened with saline water and then rubbed over the mobile phone, including the screen, the keyboard, and also the back cover. And then those samples were immediately sent to the microbiology lab for further processing. In the lab, we used nutrient agar, and we cultured them in 37 degrees Celsius for 24 to 48 hours. And after, then we identified those bacteria based on the colony morphology, gram staining, and biochemical tests as per the conventional methods. Then we also acquired our ethical approval from the ethical committee of our university, and also written consent was acquired from the participants, because our aim was primarily awareness and not judgment. So here, let's see what our studies revealed. Looking at the participant profile, we can see both female and male students took part in this. And then when we looked at the mobile phone, frequency of mobile phone cleaning, we can see that about 93% admitted that they rarely clean their phone. And only 15% said that they disinfected it daily. And then when we looked at the usage behavior, the results were even more surprising. Nearly 47% people use their phones in while eating, like in the cafeterias or in their rooms, in their kitchen, and more than 57% admitted that they used it in the washrooms. So even before our lab work began, we already had clues that behavior might be the root of contamination. Now let's see what our cultures reveal. Here is a graph representing all the types of, the major types of bacteria that we isolate. From 40 samples, we recovered total of 71 isolates. among which we can here see Pseudomonas species was found the most, about 40.8%, then coagulase negative Staphylococcus about 29.5%, followed by Bacillus species, Klebsiella species, Staphylococcus aureus, Citrobacter species, and even E. coli. So from our study, we can see that our findings mirror global studies, which suggest that mobile phones can be a silent carrier of pathogens. We saw Pseudomonas species was found the most in our study, which is because it's an opportunistic pathogen. And they are really highly adaptable bacteria which can grow even in dry as well as moist places. So by opportunistic pathogen, I mean when a person's immune system is compromised, immune system is low, they take opportunity to invade the person. Usually they are present in our surroundings, but they usually don't infect the healthy person. Especially in healthcare settings, Pseudomonas is the major cause of wound infections and healthcare-associated infections such as pneumonia or catheter-associated UTI, etc. The second most found was coagulase negative staphylococci. Even though it's found in our normal skin flora, but it's also one of the major opportunistic pathogen that can transmit from our phones to our hand and back to our phones. We also found a Bacillus species, which is also a highly adaptable bacteria. It can survive in harsh conditions because it's a spore-forming bacteria. In occasional cases, we also found Klebsiella and E. coli, which suggests that there could be possible fecal contamination, which likely points towards the fact that many of our volunteers use their phones in the washroom and maybe due to proper hand hygiene. So, next is how do we prevent it? How do we break the chain of contamination? For this, there are some simple everyday habits you can adopt. The first and foremost is hand hygiene, then disinfecting your devices regularly, 60 to 70% alcohol, avoid using phones in high risk and dirty zones, and making phone hygiene a part of our daily habits. Now this brings us to my final takeaway that mobile phones can harbor opportunistic pathogens. These organisms can easily transfer and can create a silent cycle which can sabotage the very hand hygiene we prioritize in practice daily. But the good news is prevention is simple. With consistent hygiene practices and mindful phone usage, we can break this cycle. So next time you sanitize your hands, remember a clean phone can complete the hygiene cycle. With this, I would like to conclude. Thank you so much.
Thank you very much. You have tried your level best. Good work done.
Thank you, sir.
But I would like to say that the method of sample collection was not very good. Number one. You have not at all collected the sample in a quantitative manner. Suppose there are 5 students who are standing there, you give swabs to these 5. They will collect the sample, they will rub the swabs at different places in different size of areas. And secondly, you have inoculated the samples on just nutrient table.
Sir,
You cannot do morphological characterization on nutrient table.
Sir, we also used McConkie and blood agar, and also we performed some
But you have mentioned only nutrient table. Okay. So suppose there is one colony of coagulase negative staphylococci on mobile. You have isolated. You will say yes, it is positive. You will take it into positive concentration. When it is of no significance until you know the specified area, size of the area, on what size it was. You should have done the quantitation. Okay. You should have done the sampling on a specified area. That's very, very important. Second, establishment of contamination zones. You have established 30% here, 27% there. How did you identify that this organism came from canteen, this organism came from washroom onto the mobile phone. So that was not a standard method. Okay, if some students say that I use most in the washrooms, I use most in the canteen, it does not mean that organisms are coming from the canteen or organisms are coming. So the study has many lapses, but I will say good hard work is done,
Thank you, sir.
needs further improvement.
Yes, sir.
Okay, and I would like to refer to one particular study I have gone through. That's a very big study, American study, where they have established by doing epidemiological typing that the organisms which were present on the mobile, the same organisms epidemiologically were found on the mobile phones of the doctors who visit from one bigger hospital to the smaller hospitals.
Yes, sir.
Because there it is a trend. Here in India we can work only at one place, we cannot work at many places, but in Europe, in America, those doctors can go to multiple institutions frequently. So after working here, they will go there. They will carry the organism from here to there. So just blaming the mobile as a format which is responsible for transferring the organisms.
Sir,
You
I
need
think
to
mobile
establish it further.
phones
And the identification also. Identification you have done biochemically. Okay. These days, biochemical identification is there definitely, where we cannot do anything else. But that is not 100% specific.
Sir, I would like to say that usually mobile phone hygienes are always overlooked. We take care of our hand hygiene, but after the hand hygiene, we touch the same screen which may contain this bacteria. So that's what our study aimed to raise awareness about. We should also take care of the mobile phone hygiene as well as parallelly when we take care of our hand hygiene.
Yeah. And you have just suggested that we should go for decontamination of mobile. But you have not told how we will decontaminate our mobile. Mobile is an electronic item.
Yes, sir.
Do you suggest any method to decontaminate mobile phone?
Sir, we can
Which
use.
do not damage the mobile?
Sir, I think using like 60 to 70% isopropyl alcohol wipes will not damage the screen or the back cover, which I was talking about, like sanitizing the accessories as well as the screen. At least the major parts of the phone which come in contact with our hands and faces daily. So I think, sir, those issues can be those can sanitize by using alcohol wipes, which is easy.
See, contamination of mobile is a multifactorial process. It is not only that you touch the mobile with your hands. When you open the mobile and touch your ears, your skin flora goes on that.
Yes, sir.
So, do you decontaminate skin? This is the question going on throughout the world. But nobody could reply how we decontaminate mobiles and how frequently we need to decontaminate. Okay?
Sir.
Thank you.
Thank you so much, sir.
Nice point.
Habib from Department of Medical Laboratory Technology, School of Allied Medical Sciences, University of Science and Technology, Meghalaya. Okay, moving on to the next, Ms. Kiran Kumari. Dr. Prabhidas. Dr. Prabhidas, Department of Immunopathology, PGML Chandigarh, will be presenting on development of cost-effective and time-saving indirect immunofluorescence IAF assay for the detection of autoantibodies in India.
So, good morning all of you. And myself, I am in Department of Immunopathology, PG AIIMS Guwahati. So, I will be delivered the talk that is the development of cost-effective and time-saving indirect immunofluorescence assay for the detection of autoantibodies in India. So, first of all, I would like to say that how to detect autoantibodies by indirect immunofluorescence or other assays. So, we have to, the basic concept of producing autoantibodies is that autoantibodies is a type of antibodies which can be produced by self-reactive B cells or T cells, and it can be produced by the failure of our immune tolerance. So there are different mechanisms of producing autoantibodies, like breakdown of immune tolerance, genetic predisposition, environmental triggers, and epitope spreading. So disease-specific autoantibodies are also there, like if we say SLE or systemic lupus erythematosus, the antibodies against double-stranded DNA can be there for SLE patients. Antibodies against citrullinated proteins which cause the rheumatoid arthritis, and there are various neurological disorders which are against the neurotransmitter, nerve cells, or aquaporin proteins. So, if we see this cartoon, we can see that one of the our own cell antigens, that is centromeric proteins, it can be falsely detected by our B cells, which can be cross-linked by the presentations of some bacterial DNA or bacterial proteins, and B cells recognize them mistakenly our own cells protein, that is centromeric proteins, and produce antibodies against the centromeric protein, which further leads to produce autoimmune disease which is called systemic sclerosis. So, autoimmune disease are produced by various factors. Basically, it correlates with three main factors: genes, environmental factors, and which further leads to cause immune dysregulations or failure of immune tolerance. As we can see that in genes, there are main two types of genes. One is human leukocyte antigens or HLA, and another is non-HLA genes. In case of environmental triggers, there are infections like EBV, CMV infections, hormones like estrogen, and various drugs, basically sulfonamide drugs, and which leads to cause this breakdown of cell tolerance and the defects of the clearance process of the apoptotic materials, which leads to autoimmune disease. So this is the mechanisms of producing autoimmune disease. And this is my published paper, and I have highlighted in this paper that same gene environmental factors I have highlighted in my paper, that there are some risks of HLA which cause the pediatric SLE patients and adult SLE patients, which are more prone to get infected by the EBV virus, and they produce the SLE disease. So, there are various types of autoimmune disease, organ-specific and non-organ specific autoimmune diseases are there, and their number and prevalence, incidence and prevalence are increasing day by day. Because of these diseases are undiagnosed in the rural area or primary healthcare centers. So, this is important to proper diagnose in our laboratory this type of autoimmune disease. How can I diagnose? this is a to detect the autoantibodies which produce particular type of autoimmune disease. So autoantibodies detection is the hallmark to detect the autoimmune disease. So how can we detect autoantibodies in our laboratory? First and foremost important to screen the patients when the patients come into the hospitals. The clinicians is there and then they diagnose the patients, either this patient is autoimmune disease or this patient belongs to another kind of disorders. So they check the physical examinations and they take the complete history of the patients. As you can see that there are different types of autoimmune disease, they clinically present different symptoms like SLE, which can present by skin rashes, joint involvements. Similarly, the muscle involvement is there, autoimmune liver diseases are there, and neurological autoimmune diseases are also there. So clinical symptoms are good parameter before coming to the laboratory investigations. Then once they thought that these patients belongs to autoimmune disease, so they prescribe the first line investigations like routine investigations, hematological, CRP levels, complement levels. And then if they are thinking there is a renal environment, so urine analysis and biochemical test and test for infectious diseases like EBV, CMV, etc. And then comes the second line investigations, which particularly detect the specific autoantibodies present in the patient serum. So the strategy of detecting autoantibodies in our routine laboratory, mainly there are three types of autoimmune disease we are screening, like connective tissue disorders, autoimmune liver disease and autoimmune neurological disorders. All these autoimmune disease diagnosis process depends on basically three test steps. First is screening test, next is confirmatory test, and third test is to detect any kind of organ involvement. So for screening test we have indirect immunofluorescence, we have confirmatory test by ELISA or Western blot, and skin biopsy test. So these are the basic difference between the indirect immunofluorescence and direct immunofluorescence. In indirect immunofluorescence, we are detecting the antibodies which are present in the patient serum. So at present, the detection of antinuclear antibody is the gold standard method to detect the autoantibody profiles which are available in the commercially. This is the HEP2 cell line, human epithelial cell line. This is a substrate they have used. And now the nomenclature has been changed. It is called anti-cell antibodies, but it is not basically primary to focus to the nucleus. The autoantigens can be produce autoantibodies against any particular cell organelles. So they have changed the nomenclature that anti-cell antibody, not anti-nuclear antibody. And the pattern of anti-nuclear antibodies, which can be nuclear pattern, cytoplasmic pattern and mitotic patterns. These are some patterns that we can see in the immunofluorescence microscope. At the upper side, you can see the nuclear positivity. In the middle, these are the cytoplasmic positivity, as well as in the bottom, these are the mitotic cell positivity. So on the basis of the presence of immunofluorescence pattern, we can specifically say that this autoantibody is very specific for particular autoimmune disease. Next, autoantibodies against the neutrophil cytoplasmic antibodies can also be possible to detect the vasculitis, and autoimmune liver disease panel also produced. Here we can detect 5 types of autoantibodies: ANA, anti-mitochondrial antibody, liver mitochondrial antibody, smooth muscles antibody and parietal cells antibody. All are manufactured by the commercially manufactured by the different companies, and autoantibodies against the neurological panels, in which we need the mouse cerebellum tissues or cell lines. So these slides are basically exported from outside of the India, and these are very costly. So we planned the innovative idea to produce. Sir, 2 minutes I will need. So we can produce in-house. As PGI is a overburden of different types of tests, like 12,000 tests per year we are screening for ANA, 7,000 tests per year we are screening for autoimmune liver disease, 8,000 tests per year we are screening for anti-neutrophilic cytoplasmic antibodies, and 5,000 for other tests. So this is my innovative idea, how to produce in-house slides in our system. So we took the male blister rats, I sacrificed the rat, and the liver, kidney and stomachs are dissected out from the rat, and we make the tissue blocks. After that we standardize the protocols, and we coat the tissue blocks in the multi-well slides. And you can see the beautifully see, you can see this is the slides which is combined with 3 sections: liver, kidney and stomachs. Similarly, we also prepared the anti-neutrophilic cytoplasmic slides also. This is the protocol. It can be prepared from blood samples only, 3 ml blood sample, healthy blood samples. Now I have compared our in-house slides with the commercially available slides, and we found that per test we can save 400 cost, and if we calculate yearly we can save 24 lakhs. So it is a very big gain by our institute. We are already processing in our institute by our own manufactured slides. So it is I have also performed these my prepared slides as well as the commercial slides and run together, and I have found the sensitivity and specificity almost similar to the commercially available slides. The future perspectives: by the advance of proteomics and autoantibody discovering platform, to identify the novel autoantibodies is very essential, because most of the autoantibody disease nowadays remain undiagnosed. So if we identify the novel autoantibodies, then we can detect many undiagnosed autoimmune diseases also. Thank you. This is my team. Thank you all.
Thank you, Dr. Praveen. So nice presentation. So you have described lots of panels for the autoantibodies, autoimmune diseases. But before going to the diagnosis of autoantibodies, what are the routine tests we should do before doing these autoantibody panels? What are the routine tests we'll do?
What you are saying, sir? Sorry.
Before doing this autoantibody testing, what are the routine tests, other tests, blood tests or other tests we'll do?
So there are primary screening tests are also there, like complement assay, blood hemoglobin assay. So in autoimmune disorder, the low level of complements are there. So once these first line tests are coming abnormal, the clinicians prescribe the autoantibody test.
Okay. So first we'll do this complement test,
Yes. CRP level, complement test, and other hemograms also. And platelet counts will be reduced in autoimmune diseases, in some autoimmune diseases, like idiopathic thrombocytopenia is also a type of autoimmune disease. So after seeing this first line test, they prescribe the specific autoantibodies.
And you are saying that cost of these tests are also very less.
Very less.
Okay.
It is possible in resource limited settings?
Yes, sir. We have the animal house, and we have ethical clearance to sacrifice the animals also. So it is possible, sir. I approached our institute director to produce in the mass scale in our institute, so that we can supply our in-house prepared slide to all over the nations in a very minimal cost also.
Okay, thanks.
So that will be helpful for all our nations actually.
This is the Make in India type of
Yes,
innovation.
it's showing drastic, no, you save 24 lakhs.
Yeah.
You said that.
Yes.
Then if it is
Yes.
applicable to all the hospitals,
Yes,
so
ma'am.
it may affect,
Yes, ma'am. Yes, ma'am.
thank you.
Thank you, ma'am.
For the audience, I would like to say that Dr. Praveer Das belongs to my institute, PGIMER Chandigarh. That is why I am out of the judges ethically. So these two judges will decide whatever decision to be taken for Dr. Praveer.
Any question from audience? Please ask. I have seen one poster only in outside, the first poster. One madam is there. She is doing a direct immunofluorescence test. So I am asking any present in the audience, are you doing the autoantibody profile screening? Who are doing in your hospital in autoimmune disease profile screening? No one?
Dr. Praveer, thank you very much. Actually, the time is
Thank you, sir.
over.
Next, Mr. Hejam Chandramani Singh. Sir. Calling our last presenter for the day, Dr. Pawandeep Singh, School of Allied College of Allied Health Sciences, Dolphin PG College, to talk on the efficacy of non-alcoholic skin disinfectants, a systematic review and meta-analysis.
A problem in our IT system. So Dr. Pawandeep will be presenting his presentation just without displaying the slides. I hope you understand the situation.
Respected Chairman sir, respected faculty men, organizers and dear friends, I wish to thank you for giving me an opportunity to present this study, and I feel honored. The topic of my presentation is efficacy of non-alcohol based skin disinfectants, a systematic review and meta-analysis. Let me just tell you a bit about non-alcoholic disinfectants. They are used when alcohol is contraindicated in settings like neonates, mucosal sites, burns, and flammable settings. Uncertainty remains about their relative efficacy for microbial reduction and infection prevention. Agents studied will be chlorhexidine, povidone iodine, octinidyne, benzalkonium chloride, and hypochlorous acid. Let me give you a brief background of what prompted me to present this study. Let me take you back to the COVID era. That was when all of us, including the entire medical facility of the world, felt completely defeated, and there were millions of losses of lives, including my own father. Let's go back to that bygone era when the entire world was engulfed by a wave of COVID-19 pandemic. In that pre-COVID era, hand sanitizers were being used by hospitals, in operation theatres, in ICUs as a preventive measure because there was no cure. At that time, prevention was the only option. And there was a mad craze in the world to sanitize our hands. People started carrying hand sanitizers in ladies' purses, in your cars, everywhere. After the COVID wave subsided, people started forgetting about hand sanitizers, which should not be done. Because nobody knows when some new virus or bacteria could stir up the entire world. I know the theme of this conference is AI based, but in this entire disruptive AI scene, let's not forget what happened then. And I wish to pay a humble tribute to the brave paramedical staff who bore the brunt of COVID, and we lost so many lives. A word of caution. One day I saw that on a pharmacy shop, a non-alcoholic sanitizer was being sold, which prompted me to study about non-alcoholic sanitizers. I found that these non-alcoholic preparations contained mainly quaternary ammonium compounds that included benzalkonium chloride or chlorhexidine. So, the objective of my study was to systematically review PubMed index evidence from 2015 to 2025 on the efficacy and residual activity, safety, and infection outcomes of 5 non-alcoholic skin disinfectants. The things that I wanted to display here, they are still in study and the paper is still to be submitted. But we'll share with you whatever we have. The methods used were a PRISMA compliant systematic search of PubMed 2015 to November 2025. Includes randomized controlled trials, cluster randomized controlled trials, observation studies, and meta-analysis. For those who wish to know what is a meta-analysis, it's presenting the essence of a particular topic. The focus was on risk of bias. Random effects of meta-analysis were planned. Recommendations, because we have very little time left now. Literature review shows that alcoholic skin disinfectants have some major disadvantages. I think this should be of a lot of relevance because we have been using, I myself have used, people started bathing in skin disinfectants. Excessive use of alcohol-based disinfectants causes skin dryness. Alcohol, as we know, dissolves lipids and the lipid layer present on the skin, the first lipid layer starts to dissolve, leading to dryness of skin and cracks in the skin. Secondly, in clinical settings as well as laboratory settings, use of alcoholic disinfectants has resulted in accidents in which there could be burns, severe injuries. Okay, I have myself witnessed alcohol catching fire when you start the Bunsen burner with the matches. And third is alcohol toxicity. We know that skin is a very good absorbent, and alcohol, if used excessively, can lead to toxicity. There have been reports where there has been deaths of overuse of alcohol. And lastly, in contraindicated, it's highly contraindicated in neonatal settings. These are situations that force the scientists to explore and formulate newer and more effective non-alcoholic formulations. The meta-analysis suggests that chlorhexidine displayed superior residual efficacy as versus povidone iodine. Mixed findings when alcohol carriers were used. The findings were mixed when alcohol-based carriers were used. Octreotide promising for ICU bathing and bloodstream infection reduction. HOCl effective in wound care, minimal irritation. BKC mixed and concentration dependent effects. Now, a brief about the results. Many studies have shown that non-alcohol based skin disinfectants are highly effective. They kill 99% of microorganisms within 30 to 60 seconds after application on the skin. As we all recall, the main focus during the COVID era was to break the chain of the transmission, and hand sanitizers efficacy was critical in that. Moreover, they show their activity for more than 3 hours after, if the skin is not washed. Okay, if the skin is not washed, the alcohol-based sanitizers will become ineffective because the alcohol just evaporates, whereas the non-alcohol-based skin infections, they moisturize the skin and leave it nourished and healthy. Non-alcoholic antiseptics are vital alternatives to alcohol-based preparations. Aqueous chlorhexidine shows the strongest evidence for residual skin activity and infection reduction. The results depend on the procedure, the contact time, and the carrier matter used. So if you analyze the formulation, we talk about it. More RCTs comparing strictly aqueous agents are needed. So we suggest that more studies be done on this. Preliminary results suggest bias tables. I think that's it. I throw it open for questions.
Thank you, Dr. Pabandi. It was a nice presentation. So, please explain about the advantage or benefits of non-alcoholic disinfectants over these alcoholic disinfectants.
Can you please repeat your question? Sorry.
What is the advantage or efficacy of these non-alcoholic disinfectants over the alcoholic disinfectants?
Yeah, so in brief, I can just repeat that non-alcoholic disinfectants, if used consistently, do not lead to skin irritation, do not cause skin dryness, have no evidence of any kind of major toxicity, but these results are still mixed. And thirdly, fatal accidents like burns there's a huge reduction.
Thank you.
Thank you so much.
What about the cost? What about the cost, sir?
Costs are comparable. I will give you one example. If you take, and there's a new trend which I would like, which is not in the PPT, which I'd like to share, is that we are in India, so we have our own system of Ayurveda. So people have now started incorporating the Ayurveda-based skin disinfectants with quaternary ammonium compounds. So people have started using neem and stuff, and that we are based in India, so it brings down the cost of the disinfectant. And alcohol, as we know, costs. So the costs are comparable. It is not going to be. In fact, I think it's going to be more cost effective. We have not done, financial implications of the study, but more studies have been, done. But still in progress.
Just for your information, or the information of the audience, after my retirement from PGI in 2020, I joined as Director Quality Management in one of the pharmaceutical industry, which was manufacturing various types of skin disinfectants. And I have performed personally one study which is just to be published. Ready, paper is ready, it will be published, you may see that paper very soon. In that case, the basic thing was that the major problem of alcoholic, doctor has asked the problems with alcohol. Major problem of alcohol is that alcohol evaporates within 30 seconds to 1 minute, and afterwards it does not leave any of the antimicrobial effect. Suppose a doctor check the patient, 10 patients he is checking, every time, 10 times he will have to decontaminate his hands. There is no residual activity. But I have seen in person that there is a residual activity up to 3 to 4 minutes with non-alcoholic sanitizer, but I studied that was sanitizer containing active agent that was benzothonium chloride, sodium benzothonium chloride. And sodium, I would further inform you that benzothonium chloride is a component which is actively being used in cosmetics. Cosmetic creams, it is used in a concentration of 0.1%, and the literature says that this chemical is safe if it is used less than at concentration less than 0.2%. So the hand sanitizers which are using this sodium benzothonium chloride, they have the concentration of 0.1%, and their activity, particularly I have studied. I don't want to declare my results today, but very soon you will see in the paper. Thank you very much.
Thank you so much.
Thank you very much.
As we have neared the end of the oral presentations, as a token of appreciation for our chairpersons of the day, Dr. Malkit Singh, Dr. Manoj Verma and Dr. Maliga, I call upon Mr. Fakhruddin Sharma, Mr. Fakhruddin Ahmed, to felicitate them with the Gamsha. That brings us to the end of today's oral presentations, and the results will be announced later. Of course, we are not completely finished yet, our dear delegates, participants and students. We have a very interesting session. I am handing over the session to Ms. Velvedi Gunasekaran.
Certain individuals exemplify excellence and drive. The lab heroes who push boundaries, chase curiosity, and turn challenges into discoveries, who have made their education matter. Today, we are honored to spotlight a few of those unsung champions who inspire us with their passion, grit, and innovation in the lab. Let us hear a few words shared by them through these audiovisual clips. The first hero of today is Mr. Hemant Negi, an alumnus of PGML Chandigarh. He is currently a managing director at Alta Group of Pharmaceuticals Companies, Panchkula, Haryana. They have manufacturing units at Roorkee, Uttar Pradesh. Let's listen to him.
'83 of BSc from PGI Chandigarh. After passing out, I did 1 year's job as medical technologist in the Department of Biochemistry in PGI. During that time only I came to know about the scope of this degree, the other fields also, and especially the pharmaceutical marketing, from one of my seniors who were working with Glaxo at that time. So I also entered into a multinational company which was research oriented. And primarily I got selected in the interview as top candidate because of my qualification and the subjects I had. After, then in training also I did top there because of the comprehensive knowledge of microbiology, biochemistry, pathology, virology, plasmology. But all our products were either antibacterial, antibiotics, antivirals, antiparasitic also, plus other analgesic, anti-inflammatory, and blah blah. where our degree and our subjects were very handy and were very useful. I always had a great confidence whenever I intended with the medical fraternity to promote our products because of these subjects only. And thus I could become very effective as a successful salesperson for my company. I won many prizes and stood among toppers in all India for varieties of product whatever we have launched. And because of this good medical knowledge and acumen, I was put into specialized promotions of our products into various medical and postgraduate colleges in Delhi as well and in North India. And subsequently, I got also promoted to field sales officer and area manager, RSM, and in between I got a chance because of this degree to lead a team of group product management, which was responsible for the creating strategy for the successful promotion of the brands, various brands, whatever the priority brands we were given to promote. So the crux of the message is that our degree, our subjects were very, very handy in making my professional career with a pharma company. And there I was to me it was a successful journey there. Then after a routine of 17 years there, then I thought to have a kind of feeling that if I can sell, then why not my own company. So in the year 2000, we started entrepreneurship, with pharma company, and now it is around 25 years. We are doing pharmaceutical manufacturing and marketing business, and along with this, subsequently we also ventured into veterinary drugs and then Ayurvedic drugs as well. Today we have got around 4 pharmaceutical companies, marketing companies, 12 veterinary marketing divisions, 2 poultry, 2 Ayurvedic marketing companies, but we are right now promoting the market with a network of around 350 plus distributors pan India and doing work of around more than 200 plus third parties manufacturing as well. We have got around 2 manufacturing units of drugs for allopathic for human and veterinary. Second one is for veterinary drugs. And in Roorkee, Uttarakhand and Anshula Chandigarh, we are doing food, Ayurvedic and veterinary feed supplements. Here also we have got 2 manufacturing units dedicated for the manufacturing of these products. So to conclude, my message to our juniors and co-professionals that we have got a lot of scope about the field of medical laboratory, diagnostic or research, also in other allied fields also like pharmaceuticals, biomedicals, equipment and implants, and also in the various. Hello, my dear friends. I am Hemant Dheni. I am the pass out of
Dr. Anand K. He is an alumnus of JIPMA 1990 MLS batch. He is the Managing Director and CEO of AIIMS Diagnostics, India's leading diagnostic chain across 30 states. This 25 years of work experience in healthcare and diagnostic service has won him many awards, including Leader of the Year, Award at the LH Healthcare Summit 2023 held at Dubai. He has a doctorate in business administration from Swiss School of Business and Management, Geneva, and Dr. Anand is on the screen.
Greetings, my dear friends. It's my honor to address the delegates at the 6th National Conference at ICMLS happening at AIIMS Guwahati. My name is Anand K, and I am currently working as a Managing Director and CEO of AIIMS Diagnostics, formerly known as SRL Diagnostics. AIIMS has the largest network of labs in the country and one of the leading players in this field. I am proud to be a medical laboratory professional by education, and I am blessed that I continue to be part of this community and contribute to the same in my own small way in the last 30 years. I completed my BSc in Medical Laboratory Technology from JIPMER Pondicherry in 1993 and went on to work in a hospital in Chennai, but later did my MBA in Management and entered Sales and Marketing. I worked in a few in vitro diagnostics companies for about 10 years and later joined the lab services industry as a regional head in Metropolis Healthcare and served for 11 years, where my last role was COO. Later, I was one of the co-founders and group CEO of Newberg Diagnostics, an international diagnostics alliance. In 2020, I joined AIIMS Diagnostics, then called as SRL Diagnostics, as CEO. Medical lab professionals are at the forefront of healthcare. Greetings, my dear friends. It's my honor to address the delegates at this.
Dr. Sudhir Kumar Sharma is an alumnus of PGI Chandigarh.
Good morning, friends. I'm Dr. Sudhir Kumar Sharma, Director and co-founder of AIIMS Bio Private Limited, Chandigarh. First of all, I would like to thank the organizers of ICMLS 2025 to make sure that I am present with you guys through this video message, and a very special thanks to one of my great friends, Dr. Chandra Prakash Pandey, for organizing this. Today I want to share a story of curiosity, grit, and a single-minded commitment to translate science into solutions that ease human suffering. My journey began in class 7 when my grandfather actually gifted me a book on inventions of the world. That small gift opened a world of questions and experiments, and it planted a simple belief: ideas, when pursued with discipline, can change lives. That early spark shaped every choice I made afterwards. I trained myself in clinical laboratory sciences with the BMLS from PGI Chandigarh. In those days we used to call it BSc Medical Technology, where I learned the power of technology and the quality testing in human research. I fought to sit in the master's entrance exam which I had to actually fight for almost 3 years. But ultimately, I gave the exam and then I topped the exam to earn one of the 3 seats which were available for grabs that year. I went on further to complete my PhD and a couple of postdoctoral research assignments in Canada and US, each step sharpening my scientific rigor and resolve. I returned back to India and I joined Dr. Reddy's laboratories in Hyderabad in their NCE research program. I contributed to 5 patents and also advanced 2 molecules to phase 2 clinical trials under the US FDA IND. Afterwards, I moved to Panacea Biotech, where I spent 15 years with Panacea, moving from NCE research to vaccines and biologics, and ultimately serving as the Chief Scientific and Operating Officer. Just before the pandemic, the COVID pandemic struck, I moved to Belgium, and I worked with a company called Universals, and I was the Chief Scientific Officer for Quantum Biosciences, which is one of the subsidiaries of Universals, where I worked on a fully robotized mRNA drug substance manufacturing platform. And till today, I continue to work with Quantum, but now in the capacity of Chief Scientific Advisor. I returned to India and I founded AIIMS Bio, which is a research and innovation-driven company focused on precision medicines. Our priorities these days are to work on mRNA, where we are working on in vivo mRNA CAR-T therapies, which is different from the autologous CAR-T therapy, and also we work on mRNA-based therapeutic cancer vaccines. In addition, one of the vertical we also work on precision nutrition. We have programs for cancer and tuberculosis, particularly looking at the food for special medical purpose. Every project is guided by one question: how can we move from lab insights to measurable benefits for the patients, which are faster, safer, affordable, and accessible. I owe much of what I have achieved in the foundational training at PGI during the BSc MLT days and to the mentors who taught me to pair curiosity with discipline. If there is one lesson I can leave you with, it is that stay focused, be persistent, and let dedication to patient guide your work. I wish you all a successful and fulfilling conference. Thank you all, and Jai Hind. Good morning, friends.
Unfortunately, we don't have the AV for Dr. Suman Datta. He wished to convey his regards and wish for the ICMLS 2025 to be a grand success. He has sent his message. Dr. Suman Datta is an alumnus of PGMR Chandigarh. He started his career from PGMR Chandigarh as MLT. After putting his 6 years of quality service, he joined as senior chemist in Andrex Pharmaceuticals, and thereafter soaring up to the position of quality assurance and quality control manager at Boston Scientific, Valencia, California. The prestigious Director of Biological Safety and Intuitive Surgicals, California, United States. So these heroes truly inspired us, isn't it? This brings us to the end of today's session. as we break for lunch, sharing with you the information that the valedictory function will start post-lunch at 2 p.m. Please do all join us. Have a filling lunch, everyone.